So, we'll give it a shot.
Okay. Yeah. I might have to jump in if things don't go a little too long.
Okay, so I'm going to take you through things for the upcoming October 9th. It'll be similar for October the 18th if you are writing it later on, but this is really designed specifically for the October 9th, everything that I'm going to cover for you. I'm writing it for the October 9th group, but it will be very similar for the 18th, but I'll probably have to post something a little bit different for that. I hope everyone's good. You're all good? All right, just a couple of really quick notes. So it is going to be a full 60 minutes. I doubt anyone here is going to need all 60 minutes, but we do want you to have like a little comfort zone and a little bit of a buffer in case. So you should never have a term test where you're scrambling to write and finish in the course of 60 minutes. Your test should always have like the ability for you to check things over. But it should also not be so much time that you second guess yourself. So that will also be something for you to think about. So it will be 20 multiple choice, choices A through D. And by the way, if I'm not mistaken, you guys all have a copy of this as well, right? Yes. It is available both PowerPoint and PDF? Yes. Okay, perfect. And again, there'll be short 5 one-line responses, and you guys all have access to last year's term tests. Yep. So you know what I mean by like a one-line response. Like literally it's like one word or one small phrase. So there are indeed nodding heads around the table. Okay, so awesome. And then one short paragraph, which is designed to be fairly short and nothing too involved. Do you have any questions about the overall format? No questions. So the one thing, some of you have not finished the quizzes yet. I know some of you are in the middle of studying. They all disappear on you at 12 noon on October the 9th. So you probably want to get going on them if you haven't done them yet. And if you have, I will be reposting all of the quiz questions anyways, sort of as one large PDF file, so you don't have to worry about going back and checking things through. 2 to 3 questions from the multiple choice are based on these quiz questions, so they are kind of worthwhile going over. I think I had mentioned that in class. Right? Everybody looks so serious. Okay, everyone's so serious. Okay, so there is a 5% group activity after term test 1, and this is the same test that you've just written. You'll be doing it in groups. So what will happen is that if you're writing on October the 9th, you'll be going into either EX300, 310, or 320, and then you will write it over the course of 60 minutes. Again, we don't think that you'll need the full 60 minutes, so you'll go from 410 all the way to 510. We will take a quick 5-minute break. Don't disappear, because you still need to do this part of the term test, and then you can reorganize however you want. You can just stay in the same room if you want, and whoever's near you, you can like form a group of 4 or 5. If you happen to have, like, you guys will be in the same room because your last names are the same, but if you happen to have like friends that you want to arrange to meet after in one of the rooms, you can go ahead and do so. So we will give you time and opportunity to rearrange. We want it to be like a fun experience, I guess. And again, it will be sort of the same version of this. How are things going on the stream? So seems to be okay? All right, awesome. So I will have office hours on Tuesday. I have class from 10 to 12, but outside of those class times, I will pretty much take appointments all day during regular office hours from like 1 or 12:30 to about 5 o'clock. On the day of the term test, there will be a special study space here in New College. And Wilson Hall 2007D is just on the other side of the stairs over here. So it's like a bigger room. So it's a long table like this one. It's a bigger room, lots of windows. It's a great study space. There is like a TV set up in there as well. So if you need me to go through a concept with you, and we want to make sure like it's a relatively quiet study space. But you can stay there as long as you want, and then we're going to leave at about 3 o'clock to head over to EX, or you can stay and do whatever you wish. All right, so that's available for all of you, and we are just trying it. I've never done any of these things before. As I mentioned to you earlier, your class, we want to try a few different things, and we want to see if it's useful for you. If it is, we'll do the same thing on the final exam. Just creating like a special study space on the day of your exam. All right, any questions so far? No? Yeah, Farbad. So the room is slightly bigger than this one here, and it's actually, yeah, so we want everyone to at least discuss it, and it's really hard if you have like a group of 6. So we usually recommend groups of like 4 or 5. If you really want to have like a group of 6, like you're really tight with like 5 other people for a WOD, we're not going to say no, you can't do it. But it is like really hard for 6 different people to have like a good, meaningful discussion. Like, oh, I didn't get that for question number 1. Like, what were you thinking, and why did you choose that one? And it's harder to do that. So, yeah. Does that answer your question? You can't have a group of like 20.
12,
Yeah, yeah. 12 would not be good. Yes, Yuan?
So after the, if you're taking the test somewhere else,
Yep.
what would be the format?
Yep. So if you are writing it somewhere else and you need accommodations, we also have a separate activity that you can take away with you, and then you'll write it up. And it's a reflective activity. And what we're trying to do with this post-term test is to get you to think a little bit about, oh, why did I choose that answer? And so it's similar to get you to think about how you prepared, how you studied, or like, oh, I didn't even know we needed to know that figure. And there it was. So you won't be losing out on 5%, in other words. Okay, so lecture 1, to be honest, if you have been spending the most amount of time on lecture 1, stop studying it. So it has the lowest amount of coverage on term test number 1, and it's mainly multiple choice test questions, and I can confirm for you that you only have like 3 multiple choice test questions and like 1 short answer. It's really, it's pretty straightforward from this section. We didn't spend a lot of time on it, so if you are confident in lecture number 1, you should really stop. Like, that's it. Have you all finished studying lecture 1?
Yeah.
Okay, good. So done. One lecture out of the way. As you'd suspect, like this is a diagram because we covered it both in lecture 1. We also covered it in lecture number 2. This diagram is one that you should probably spend some time on. I think most of you have done the quiz questions. As I mentioned earlier, there are a couple of quiz questions that are very similar to what you'll get on term test 1, and here I can confirm that it kind of revolves around what you have already learned about. And you already know about Borrelia and everything else. So it's a good idea to make sure that you go back over this one slide, but other than that, I don't really want you to spend too much time on this. So I'm mentioning this because a lot of times students are just wondering, like, you know, do I need to memorize these definitions? I'm not huge on definitions, so I think like the short answers, not, you know, most of them are not like definition type of questions. But dysbiosis, I expect you to know what dysbiosis is. So if it comes up in a question, an individual is experiencing dysbiosis, you know that they're not in their normal health state, and so that there's something wrong with the gut microbiome. And you don't need to memorize like anything on this slide, but terms like dysbiosis, it's more likely that you'll just see it incorporated into the question, okay? So I don't want you to have this false belief that, oh, he's going to ask me for a formal definition of things like dysbiosis, all right? Cindy, you look like you're shocked. No?
No, this is my like this is my resting face.
Okay, it's your resting face, okay. All right. I'm also going to do my resting face now. Nice. All right. So as I mentioned, and I just wanted to confirm with you again that you are not responsible for this part of the slide. In case you were wondering, there are no questions about anything on here, over here on the left, on the right. So if you were thinking that I was going to sneak in a question from that, you do not have to worry. I will say that it is probably a good idea to make sure that you know the bullet points that are here on the left. And as I said, there's not very much from this particular lecture that you should be expecting. I've already indicated one of the figures, the earlier one, and I've also indicated that this slide is one that you should probably know as well. All right, so again, I just want you to really use your time effectively. Again, I also mentioned to you earlier that you do not need to memorize all of the numbers here on the right side of this diagram. and that the most important part is that throughout the GI tract, as we go from the stomach, through parts of the small intestine like the duodenum or duodenum, jejunum, and ileum, that we see an increase in both the number, so here we're talking about number, and then we're also talking about diversity. So we get more and more bacterial species, and especially as we go down into the colon. So put it to you a different way, because this sounds like really simple, and how could he possibly make a hard question? Do you remember one of the measures that we learned in the last lecture number 3.5/4? Do you remember what that diversity index was called? So the face diversity index, do you remember that? You don't remember that? I swear I covered it. I swear I covered it. Okay, so another way I could ask this is like, oh, in these different areas, instead of just asking you like for a number, where would you expect to see like a higher face index? And you would have to know that. And so I want you to start thinking about how to integrate the information from lecture 1 into like lecture 4. I hope that's fair, and I hope that's something that you can understand, like why we want you to do that. Okay, and I think, and I just also wanted to confirm before we start printing off like 300 copies of this term test, that you guys want to sit like farther back? Is that going to hurt your neck? It's up to you. Like, you're welcome to sit there. I just don't know if that's like too close for you. So you don't need to memorize all of the different bacteria that are here within the colon. Okay, so I just wanted to emphasize that. I'd highlighted sort of the lactobacilli, but the lactobacilli is straight through the entire GI tract system, and it kind of pops up like everywhere when you look at the gut microbiome. All right, so again, don't spend too much time on these things. Sorry, I'm going to take questions at the end if that's okay, Yuan. So if you continue on, like this is another slide that I'd like you to maybe go back and revisit, because I know I talked about this in the last lecture where we talked about enterotypes, right? And then I reinforced the fact that, oh, do you remember from lecture 1 that we actually talked about the bacteroids, the Prevotella and the Ruminococcus, and then we saw how the ratios of these actually did change in different health statuses. So I wanted you to go back and revisit it. And again, it's unlikely that on the term test this year that you're going to have a straightforward definition where like, what does it mean when you have like an enterotype, or what does an enterotype mean? But it might be incorporated as part of a question. Like, a person sees a change in enterotypes or a change in diversity, then you know that things are being affected. All right, so we finished lecture 1. So if you do have any questions that remain outstanding for you from lecture 1, now is the time to ask away. Yep.
So Bates index represents the diversity, and so there would be a higher Bates index in the colon, correct?
Sure, yes, that's correct.
Okay.
Any other questions from lecture 1 at all? Yep.
Is enterotype another way to say diversity?
It's not really diversity. We're looking at these 3 major genera. So we have these 3, and we're just seeing a difference in the ratio. So does that get reflected in changes in diversity? Yes, but we're not really talking about enterotypes as a measure of diversity. Right. So good question, Cindy. So yes, it would apply mainly to humans. Rats have a very different enterotype than we do. Any other questions? Sorry, who is that? Lauren. Yes.
So when you say the rats have a very different enterotype, if they were to start eating more of a high-fat diet, they wouldn't have the same changes as us, or do you expect them to have similar changes?
So that's an interesting question. So when we do so the experiment that was done in the paper that we covered with Parkinson's, they actually transplanted a human microbiome into like a rat sort of background. There they actually did develop like a human enterotype. And we can change that enterotype, the ratios, through diet, through disease, and that can also be reflected as a change in diversity. So.
Do we expect rats to exhibit the
Uh, in general, yes, yes, yes. Any other questions? No. Okay, so we'll jump to lecture 2 then. Are there any questions from the online group? No?
Yeah.
Is there anyone online?
There's a couple of people.
There's a couple of people online. Couple of people online, if you hear me and you have questions, feel free to ask. I'll probably not answer them right away, but we'll get to them eventually. All right, so lecture 2, heavy coverage on term test number 1. So this is the one that if it's been a while since you've studied it, you go back and study it on the weekend. Like for me, what I would recommend you do, spend the weekend studying like lecture number 2 and lecture number 4. If lecture 3, some of the concepts were challenging, like you would probably do that and get that all done over the weekend. Make sure that anything that you're not clear on, you come back and ask me like on the Monday or Tuesday, last chance Wednesday, and then I think it's fairly manageable. Like that's just my opinion, but I don't know what other things you guys are preparing for, so I don't know. You don't look all that tired to me, so okay. But on the inside, you're tired. Okay. This is also like mainly multiple choice, but there's a lot of multiple choice related to this. So different things that are on the bullet points that you see here, I do expect you to know some of these bullet points. So we also see changes specifically in different types that are human related, like actinobacteria. You should know like the overall changes that happen as you change your diet to one where you're no longer metabolically healthy and it's no longer in balance. So you're going to see a decrease in bacteroidetes and an increase in firmicutes. That should be the common trend. Anytime you see something bad happening, so a change in exercise, not exercising more, you'll see the same change. Increasing the caloric intake to increase obesity, you'll also see a decrease in bacteroidetes and an increase in firmicutes. Okay, so those are the changes that you should be keeping in mind. Actinobacteria is another one. And again, now you should have a better understanding based on what we covered in the last lecture about how the shift in the bacteroidetes and an increase in firmicutes might actually lead to changes in short-chain fatty acid production. Okay? All right, so again, there's not much here other than to go back and kind of emphasize that changes in diet will cause the same changes that were outlined there. Changes in exercise might change in exactly the same way. And here we're looking at changes in specifically related to high-fat diets. So you should know like bacteroids kind of dominate. Carbohydrate-rich, you will see more of the Prevotella dominating. And generally speaking, those are the different types that you should probably know for the term test. Is that a surprise? No, hopefully not. Okay. And again, I don't know what your expectations are for the term test. It's straightforward. There are a couple of questions where you have to think your way through them, and you have to integrate different concepts. But I'm kind of mentioning like how you integrate concepts in this review today. Yes. Hi. Yeah.
I forget that there's some questions that I sort of didn't expect to be there. So would you say that?
Can you give me an example?
There could be something, or would that be like identical content from last year?
So no, we did not cover the same things last year. We covered about 75% of the same material. So you'll see a lot of the same types of questions or the same concepts. But we also talked about other things last year.
So if there is a one question that I have just no idea about, it's okay. From last year.
Well, I can't say.
Well, like, is it?
If there's a choice that you've never heard of before, then probably. Yeah, because we did talk about like different things. For example, I think I told you we didn't talk about the human study with autism. Like if you saw an option talking about like movie prep, like last year's class would have like no idea, like they wouldn't know that. Your class does. In terms of like vancomycin, like your class would know what vancomycin might be used for. Last year's class would have like absolutely no idea. So yeah, I think there are some differences. I would and that's one of the reasons why I don't really like putting up previous tests. It's more to get you familiar with the style. Yeah, and like we try to keep it fair each year. So, yeah.
Because I see questions like at OSB and like CRAB.
Delta Phos B? Yeah, so we didn't cover Delta Phos B, CREB, or any of those pathways right now. So.
So it's okay if you don't know about that?
No, it's not okay because you should have had that in your second year classes, but it's okay for the upcoming term tests not to know it. Like if you don't know it because like did we learn this in class? You did not learn it in class, so that part is okay. The fact that you don't know them, that's troubling for me. Like you should have learned, if you've learned anything about addiction, then you should have learned about Delta Phos B. Like Delta Phos B is like the molecule in addiction. If you haven't learned that, then that's a problem. Well, you should have learned it. I'm just saying. We'll come back to that later. Let's focus on the term test right now. Okay. So again, here you should know things like meconium. As I mentioned to you in this class, the basic sort of concept is that in utero, we're going with the model that it's sterile. Like there's nothing in there, so you would expect to see like nothing in the meconium. You would expect to see nothing in the amniotic fluid, and that's what we're going based on, okay? And I mentioned that in class. I told you also that you do not need to know about Hirschsprung's disease and aganglionic megacolon, and you don't need to worry about this last bullet point. All right, I still have some more slides. Okay, so will you get questions from this slide?
Yes.
Yes. Right. So I can tell you I use, hi, how are you? Oh, wow, you guys just, wait, did you just swap out with Jeffrey?
Yeah, Jeff has something to do.
Nice, nice. Did you give each other a high five when you swapped out?
No, but we
No.
should do that next time.
Okay, next time. Okay, awesome. So in utero, we're going based on it being sterile. I do expect you to know the differences between cesarean section or C-section and then vaginal delivery, where the initial sort of few weeks of colonization, we would see more of one type or groups of bacteria versus the other type. Eventually, they all become enterobacteria, and eventually they will continue over the course of like 1 to 6 months to redefine themselves. And eventually we end up with these stable bacteroides and these spermicutes. So any of the things that you see up here, like increase in immunoglobulin G, decrease in toll-like receptors, like these are not ones that you need to know, nor do you need to know anything over here on the right, although we just talked about things like obesity, high-fat diets, carbohydrate-rich diets, which don't actually cover bacteroids, spermicutes, but more of the Prevotella. All right? So is there anything that you guys need to confirm now or just want to double-check on? Yes?
I wanted to ask, if bacteroides is the main carbohydrate-utilizing bacteria, why would it dominate in a high-fat diet?
So a lot of the metabolic breakdown products actually get converted. So it's not necessarily the ones that you'd be thinking of. And we'll come back to that in the last or second last slide today.
Okay, so it's like that next step.
Yep.
Okay.
Yep. Yeah, Cindy?
What about like the different allergy antibiotics?
So anything over here on the right, you don't need to worry about. No. No. Yep.
You were talking about the shift in bacteroidetes and then the increase in firmicutes and how that affects short-chain fatty acids.
Yep.
I was wondering if you could elaborate on that.
So that's actually from the last lecture. And so I'll cover that really briefly. And there's a really nice diagram that actually shows like what different types of bacteria cause the conversion of these different short-chain fatty acids.
Okay. I thought you covered it in lecture 2, so that's why.
No. So I can also confirm that you will have a couple, but not very many questions related to dynamic novelty. And I can also tell you that you should be familiar with the concept, and this is related to dopamine. But any of the slides that you don't see in this slide deck, so if you're wondering, like, am I going to put the different types of drugs like cocaine, alcohol, barbiturates, THC that affect the BTA nucleus accumbens pathways. You don't have to worry about that. If you look on those diagrams, by the way, there is like delta phos B, just saying, delta phos B is all over the place. And you don't have to worry about the circuitry of like dopamine release. You should be familiar with a couple of things. So some of the models, like you might find them useful and helpful for you on term test number 1, and how dynamic novelty can be used to take one of the groups that is obesity resistant to have that extra sort of difference that allows them to start eating more food. Why that is sort of important in terms of dynamic novelty. It is one of the tests that we can use that the dynamic novelty can actually induce a change in feeding behavior. So why do people change their feeding behaviors? Sometimes it's just like dynamic novelty. So why don't you buy like the new iPhone 11 that has like 3 cameras? Well, it's the same black color, but if there's like another extra thing, like it's a green color with 3 cameras on it, like that might be just enough to push you over the edge to pay that $1,600 for that iPhone 11. It is something that you should be aware of, but I think, to be honest, like this will be pretty straightforward, although you'll have to tell me like after the test if it was a straightforward question. You should also be familiar with what progressive reward models mean. Does anyone want to give me like a quick 1-2 sentence explanation for how we examine progressive rewards? Anyone?
With the bar leader test, so like if you increase the level of difficulties, but like decrease the reward, they'll still press it the same amount, like induces similar to cocaine, something like that.
So do we actually reduce the reward?
Not really reduce the reward, but like keep it, like it's harder to obtain increased difficulty for the same level of reward.
Okay. Yes, did you have something different?
You make the challenges more and more hard, and you see if there's a threshold at which the rat's like, oh, I'm not doing this anymore, it's not worth the reward.
Right. Okay, so I think if you combine those 2 answers, that would be perfect. Right. Awesome. Okay, so that was sort of it for that. If you know these 2 slides and you can kind of work your way through them, I don't think it's a challenging concept, to be quite honest, but again, that's me. Then you should be fine for this section. And by the way, some of you are wondering, like, are these the only slides that you need to know? What do you think? No. Actually, kind of. Everyone's like, huh. Well, that's up to you. Like, you can either decide that, yeah, okay, or you can decide to just do your own studying thing. But it's hard because it's like ripping like a chapter out of a book, and it's just like, learn this chapter. It's better if you understand like the rest of the lectures, but these are the main slides that you should be aware of. These are the slides, in other words, that I use to make your term test. So Parkinson's disease, a couple of things. You do not need to know memorized numbers, but you should be aware of like overall trends. We do not know the vast majority of the reasons why people develop Parkinson's. The majority we just don't know. We can't attribute it to a genetic mutation. So when we go back in and examine someone who has had Parkinson's, we really have no clear genetic link. The one thing that we do know is that alpha-synuclein seems to be really important, and we know that this misfolding of alpha-synuclein is a key step. We see it in a number of other diseases like synucleopathies. It's not just Parkinson's disease, but we also see it in multiple system atrophy, which means that multiple areas in the brain, from the brainstem, cerebellum, etc. And I didn't cover that, so you don't need to know all of that right now. And Lewy body disease are going to be affected. So this slide kind of puts most of that in perspective for you, that we start with alpha-synuclein monomers. So we all have alpha-synuclein. We have it in a number of different places in the body, the brain, the gut. Many other tissues have like alpha-synuclein. for whatever reasons, maybe due to phosphorylation, and what residue might you see phosphorylated? So if I had alpha-synuclein, and I believe that phosphorylation is the key step in actually causing it to aggregate and start to misfold, which serine residue would you most likely pick? Yes, for which one?
129.
Okay, great. So I was a little worried there for a second. You do remember that one, right?
Yeah, phosphoserine 129.
Right. Okay. So we think that that's one of the key steps. The phosphorylation might eventually lead to protofibril formation, fibrils, and then eventually, like the thing that we often use in many cases of Parkinson's. Not the only one, because obviously there are other diseases like multiple system atrophy and Lewy body disorder. But this is one of the hallmark features of Parkinson's, is the collection of these Lewy bodies. Okay, so it all begins with monomers, misfolding. That might be related to the level of phosphorylation, especially serine 129 on alpha-synuclein, and that will lead to increased protofibrils, which then form fibrils, which then collect even more and aggregate as Lewy bodies. Out of all of these things here, from the left all the way to the right, which one would we expect to see more of in the insoluble fraction if we were to use Triton? So here on the right? So we would see, this would be in the insoluble fraction. We probably see some of this, and most of this would be in the soluble fraction. Does that kind of put that in context for you, and does it still make sense for you? So this is soluble, this is insoluble when we're trying to use Triton as a detergent, and this is sort of the continuum. So this is all soluble, less soluble, even less soluble, and this is mainly in the insoluble fraction. I should have used this diagram to explain that in the first place. Right. Okay, so you don't need to know specific details like what ages, etc. I am going to say that you probably, I may not have this in the right order. Okay, so it's on this slide, but it's in a slightly different way. Do you remember what else Parkin is called? Yes. Pink one is actually related or a binding partner for Parkin, but do you remember like what? Does that ring a bell for everyone? So the ubiquitin E3 ligase. There's also like pink one, but that's sort of a minor form of it, and it's also involved in the Parkin pathway. And then alpha-synuclein. So these are the main genetic causes that make up like 10% of all of the known causes of Parkinson's disease. So loss of dopaminergic neurons usually are accompanied by melanin. And to be honest, you do have a question related to this slide. I'm just going to put that out there. So if you're writing on October the 9th, it's probably a good idea to just go through this slide, make sure that, I'm hoping most of you have seen these before, but you'll see something related to this slide as well. You do not need to memorize this slide here, but you might see as sort of an indicator. So if you're given some different symptoms where a patient has bradykinesia, I'd expect you to know what bradykinesia is. I'd expect you to associate this with Parkinson's disease, and I'd want you to associate it with the rest of everything here. All right. All right, see you later. Okay. All right, so again, making sure that you know like the definition of vaccinic. So last year they didn't have this definition, so just to kind of put that out there. And I actually come back over parts of this slide in lecture number 3. But here I put it up there for the definition, so please make sure that you go over that. All right. right, and we're almost at 2, so let me speed up just a little bit. And there's no questions online yet, right, Michael? Are they still there?
Yes, we still have 15 watching. Oh, never mind. A question just came in.
Oh, a question just came up.
You mentioned Lewy body disease. Isn't an introduction of Lewy bodies into the brain an indication to Parkinson's? What is the difference between the two diseases?
So there are a number of other conditions where you can actually see Lewy body formation, and you can also, it's hard to say because the paper, I'm not sure if they're actually referring to a specific paper because they're not true prions. So prions, if you were to inject prions into a brain, you can actually begin the process of seeding. It's very, very controversial as to whether or not seeding, so a misfolded Lewy body containing like misfolded alpha-synuclein can actually seed normal alpha-synuclein to start to misfold and then cause a cascade effect. That's been very controversial, and I'm not sure if they're referring to that. So yeah, if they want to come back and discuss that in further detail, I'll be happy to do that with them. All right, so thank you, online group. Okay, so lecture 3 has a lot of coverage on term test number 1. You will need to know this lecture for the short answer, okay? And you'll have to use like bits and pieces from like different lectures to make your answer a little bit better, but I promise it will not be like a really impossible question. And I do know that some of you spend a little bit more time writing than others, so we have it planned that most students will finish writing by about 40 minutes, and then checking over for like 5 minutes, and then you can continue on if you want, but we're hoping that some of you will be able to finish. And I guess we'll see if our prediction is correct. So do you think that this is on your term test? Do you remember going over this in class? Yeah, so do you kind of remember this appendectomy controversy? So some people say yes, some people say no. It's probably a good idea to be familiar with the fact that appendectomy is controversial, and that some studies say yes, some studies say no. But you'll probably see at least appendectomy on your term tests. So I didn't want it to be a surprise. I know not everyone has the same physiology background, and not everyone may know what an appendectomy is. So I just wanted to make sure that if you see that word and you're seeing it in relation to something we've talked about, this is sort of what it's referring back to. So again, we've already covered this slide. I've already mentioned that you should probably know this slide. It will be on your term test. You do not need to know any of the structures. You don't need to know any dates. You don't need to know any names. All of these things are kind of there just to tell you a little bit more of the story behind it, but you don't need to worry about all of those. So it's just a confirmation that you don't need to know structures. You don't need to know names or dates. This slide, though, you do need to know all of the different things that are on this slide. So whether or not we inject in and introduce a toxicant like reserpine, or we inject in MPTP, or we are injecting in 6-hydroxydopamine or large doses of methamphetamine, these can cause the loss of catecholaminergic neurons, and particularly dopamine, which is a catecholamine, right? So if you've taken, for example, any of your psychology courses like Psych 396 and the neurochemical basis of behavior with Dr. Wood, you'll know that dopamine is one of the catecholamines. And of course, we had already talked about this, so you need to know this slide. Straightforward slide, but you need to know it. And you guys were all expecting that you needed to know this one, right? Yeah. Right. Okay. Again, no need to know like any of the different study names or anything else. So this isn't like a psychology course where you have to actually know the study authors and then the dates. And you're more likely to get questions where it already includes different types of things like dysbiosis directly in the question. So just more of a confirmation of what you don't need to know. You should be familiar with Brack's hypothesis, though. So Brack's hypothesis comes up, and I think last year, it was the first year we actually talked about it, so I think there's a question or two related to Brack's hypothesis in your previous term test, which you all have looked at, right? Oh, you're that group. Are you that group? Are you the group that kind of like, I need to get all my studying done, leave it as sort of a reward for myself, and then do the term test? You're that group? So,
Lose confidence.
is it? But I think again, you shouldn't lose confidence if there are terms that you aren't aware of. That was an earlier discussion. And I will probably be posting a couple of questions between now and Wednesday anyways, so it's probably a good idea to check in on the announcements page. You never know what will show up there, and there might be some useful things to think about. So I just wanted to go back and emphasize a couple of things. So one of the reasons, or one of the things that you can test, in most of the studies that we looked at, we actually didn't look at Brack's hypothesis because Brack's hypothesis is very, very specific. It says that in the gut, alpha-synuclein starts to misfold, and this aberrant or misfolded alpha-synuclein, which probably includes like the phosphorylation on serine 129, starts to accumulate in the gut, and this misfolded protein can then transfer and move along this vagus nerve up into the brain. And in the brain, it then starts to misfold other types of alpha-synuclein that is not yet misfolded up in the brain. And that's Brack's hypothesis. We have not actually studied Brack's hypothesis. We've mentioned it, and we've looked at what happens in the gut in terms of short-chain fatty acids. In a second paper that many of you got confused on because I didn't explain it well, how we can trace this vagus nerve from the gut to the brain. We can see that it actually does exist, but in none of the discussions that we've had have we gone through a paper that actually examines and then tests this Brack's hypothesis. So I just wanted to be clear on that. And we can actually, if we were to test for Brack's hypothesis, we would see this misfolded, sorry, when I'll get to you in a second. So we can see these misfolded alpha-synuclein within the nervous system that supplies the gut and parts of the glossopharyngeal and, of course, the vagus nerve. Another test that we could do is that if we cut the vagus nerve, so we cut this transmission line, these vagotomized individuals have been shown, not just by Svensson, but others, like many other papers have shown this, that vagotomized individuals where the vagus nerve that is innervating the gut and comes up into the brain, these individuals have a reduction in the risk of developing PD compared to the general population. Because we don't know like what causes PD, but they actually, fewer of these individuals will develop Parkinson's. All right, so here, you don't need to know like what the construct itself actually looks like. I'm assuming you know most of like what an ATG start codon looks like, that first methionine, this is the full Phi1, but you don't need to really worry too much about this. You should probably go back over your notes what the Phi1 promoter is used for. So it's particularly good in epithelial cells as well as in neurons to overexpress alpha-synuclein. And I mentioned the importance of 12 weeks and why we do experiments at 12 weeks as well. Do you also have class? Okay, so I'm hoping the recording and streaming is going okay. All right. So you do not need to memorize any of the overall size effects on the data, but you should probably know some of the different tests, like how do you actually test to see if motor function is being affected, so beam traversal, how long it takes to cross a beam. The worse their motor performance is, the longer it takes. If they're coming down a pole, they need a lot of motor coordination. It takes them longer. It takes them longer to remove an adhesive tape placed right on the back of their head, behind their nose, or on their nose. And then hind limb clasping as a reflex. All of these are longer in these animals, and you should know the difference. The only thing I want you to know, not the size, but where would you expect to see bigger differences? In an animal that has overexpressed alpha-synuclein that has an intact gut microbiome, or one that is completely germ-free, okay? And I think we've covered that. You need a gut microbiome to see these effects. So I won't lie. You need to know how to, or the major points that are on this particular slide as well. So what serine 129 phosphorylation represents, if we'd expect to see more serine phosphorylated serine 129 of alpha-synuclein in the soluble versus the insoluble fraction. What do we mean by soluble and insoluble? So it's soluble in Triton, or it's insoluble in Triton, which is a detergent that is used in protein extraction. All right, and okay. Yes.
Someone just asked for if you could redefine for them what X-scenic is.
I will post something a little bit later. They gave me a thumbs up online. How cool. Or is that just
I'm
you?
just giving you the
Oh,
thumbs.
never. Okay.
Yeah,
never.
they just asked for you to repeat the definition of X.
Okay. All right. So lecture 4 also sadly has a number of questions on it as well. You'll probably need to incorporate some of this in your short answer questions. So you will see like IBA1. So if you see that, what are you staining for? And what else can we stain microglia with? TLR2. So TLR2 is something we looked at in lecture 1, a little bit again in lecture 2. Do you remember what the downstream result of activation of the microglial or IBA1 positive cells that contain TLR2, what would you end up seeing in those cells? So you'd see increased NF-kappa B. What else might you expect to see? Because this again gets into integrating that information.
Increased IL-6 and TNF.
Right. So this gets into, there are changes in morphology, which you should generally know. So which ones have like thin cell bodies versus more rounded amoeboid bodies. And as they go from being less activated or in a quiescent state, we have these thin cell bodies. And then we go into being activated. We get these round amoeboid cell bodies. We have fewer branches and they get activated. So make sure that you're aware of that. And this gets into what your colleague was mentioning a few seconds ago, that we would probably see the release from these microglia of a number of different pro-inflammatory cytokines like TNF-alpha, interleukin-6. Oh, and I also put this up here for a different reason. You are free to use any abbreviations that we've used in class. So anything that we've seen in class, you don't have to write out interleukin-6. You can just write IL-6. As long as it's in the notes, then we will accept that as sort of a short answer question or part of a short answer question. It can't be something that you've made up yourself. So if you have your own shorthand form, like that's great, but if it's in the notes, then we'll accept it. So just again, when we're talking about IBA1, that's a stain or an antibody that we use to stain for microglia. These microglial cells contain a lot of different things, but they are also CD11b positive cells as well. So this, again, these are primarily microglia. So we've looked at different markers or things that are expressed in microglia. All right. And again, here you should just know what we might do and what you might expect, because we didn't go into it in a lot of detail. So this, if you're thinking about what I'm trying to tell you, on your short answer question, it's not necessarily something where I'm asking you to repeat back what we've learned in class. but maybe to integrate and synthesize something that we have started to cover, but maybe not have completely finished, or to kind of expand on it. So for example, we didn't talk here about any of the microglial sort of differences, but you could probably extrapolate like what you might expect to see. You might be able to extrapolate what you might be able to see in terms of alpha-synuclein. And why might we actually do these types of experiments? Like what is the importance of these different types of experiments, okay? So just to get you to think a little bit about that, and one other thing, on your term test, we will also be giving you, unless I'm mistaken, I'm trying to remember if I did this routinely, we're usually giving you different things like a short form like ABX, so antibiotic treated, and then in brackets like ABX. But we would expect you to know like what SPF means. Like we've covered it enough times. So if you see GF mice and you raise your hand and you're asking like what does GF stand for, like we would expect you to know what that is. We didn't go into this in a lot of detail, so if you see like ABX, we would have to give you that definition. But GF, SPF, like we would expect you to know. And you all by this time know like ASO means alpha-synuclein overexpressing, right? Right, okay. All right, so again, you should probably know that most of what the genomic analyses look at is the 16 ribosomal small r RNA sequencing. We do this to get a quick, dirty picture of like the different families or genre of the bacteria and how they're changing and how they're shifting. So it gives us almost like a snapshot of like the enterotypes, but a little bit more fine detail than just enterotypes. So it's more like a diversity index that we would be generating here. And yes, when you see things highlighted for you on this slide, then you should know the general trends. And I can't recall, but I think one of your quiz questions had something like this on the last one, right? Have all of you done all your quizzes? Yeah. Farbod. No, no. So I think this was on one of your quizzes, if I'm not mistaken. So I do expect you to kind of know that overall trend as well. Actually, I'm not sure why this slide is in here. Okay, have a good appointment. Check back for the recording later, and maybe I'll see you in office hours or. Yeah. Okay, see you later. I'm just trying to remember the reason for why I put this in here. Was there anything? There was a reason I put this in here, but I can't recall off the top of my head.
I think it's the difference between like motor and fecal function. How like this test only caused motor symptoms and didn't cause changes in fecal output.
So for this, okay, sorry, I'm just backing up. Did I ever say that? I'm just, okay, because I'm hoping not, or I put it in context. That's actually not true. If you go all the way back, and I didn't cover it, if you look very carefully at one of these graphs, which I didn't cover, they actually don't poop normally. So if you have like alpha-synuclein overexpression, they actually don't poop normally.
But do we have to know, like, because in the next experiment, then it didn't show symptoms. So do we have to know, like, that distinguishing between the first experiments where this kind of experiment showed motor?
Okay. Right. So I think the way that I explained this here, so one of the things that you might expect if you believe that previous graph that I just scrolled all the way back to and then went all the way back to this one, They actually have lower fecal output. So one thing that you might suspect is that they actually have a change in weight. They actually don't. So their weight is actually pretty close to at 12 weeks of age. Here, what is being mentioned is that in terms of the PD transplantation, they're really not seeing much difference. But I don't expect you to know, like literally I'm trying to think of why did I put this slide here.
I think it might be because of the point of like, you have to be genetically susceptible in order for the transplant to actually make a difference.
Right,
'Cause I think it
right.
might really
So I just want to be clear that all of these studies use this Phi1 promoter alpha-synuclein overexpressing mouse. So if you, and thank you, I think that jogged my memory a little bit. If you are asked to come up with a model system, don't just write mouse. Like it has to be, no, I'm serious. So make sure that it's clear that you're looking like at the Phi1 alpha-synuclein overexpressing mouse, and then if it has a gut microbiome, if it doesn't have a gut microbiome, if you're treating it with antibiotics or you're not treating. So just be like really clear in your models, because it's not like we're trying to take marks off, but if you just write mouse, like we don't know if you either never learned it, just forgot it, aren't sure, and that's all we want is some clarity in your responses. So just make sure that you're complete in your answers. Did most of you write this down? I think most of you did, right? So acid inhibitor. So the Prilosec, acid inhibition, MubiPrep, and vancomycin. These are all kind of done. These are the long-term benefits. Apologies in advance, but you have a question on your multiple choice with this here. I decided that I really liked Biochem 210. I wanted to make like a Biochem 210-ish question, so I did. But as long as you've kind of studied the major features of the ones here on the left, then I think you'll be in pretty good shape.
Do we need to know about Clostridium cluster?
Nope,
Okay.
nope, nope, nope. Any other questions about this slide? No? Okay. All right, and again, just to kind of re-emphasize, you do not need to know all of the different data trends. You're not going to be asked on the term test to analyze data, and you should kind of just know what sorts of scores are being examined, and that we generally see a positive outcome that's long-lasting in terms of gastrointestinal symptoms. They improve, and we also see an improvement in autism rating scale, so their behavior, interaction with others increases, and social responsiveness also increases as well, okay? And they increase in sort of a positively correlated way. The better their gastrointestinal symptoms get, which is what you're seeing here, the better their social interactions get. And all of these things are occurring 2 years after their gut transplantation. And this sort of is the face diversity index, so just to kind of put up that phylogenetic diversity thing. Do you think you need to know this? Yes. Do you think you need to know about this here on the right? I'd say it's a pretty good idea to kind of go over this slide. There's not a lot of data on it. It's just mainly changes, or just to outline that better diversity is better, and that after transplantation that we saw increases in certain types of bacteria and decreases in others, and that's really it. This experiment here, there are a couple of questions. So there are 2 multiple choice test questions based on some of the data on this slide. And then there's a scenario question based on this particular slide and understanding like how this all works. So there were quite a few questions in the last lecture that we covered on this slide. are you guys all comfortable with this slide now?
You want us to
Sit.
explain it to you, so to know that you fully understand it?
Oh, I don't know. You guys, if you is everyone comfortable with it? Do you understand like what we're looking at?
There's a question on the discussion board.
Yes.
About it.
Yep.
It just says, basically, why, like, so they start off without the G-glycoprotein, and then
Right.
they add RABG, and the person's like, why would they not just start with a thing that already had the G-glycoprotein and RABG?
I think I can answer that. I think it's because if you don't have the G glycoprotein, then you know specifically which cells that they were infecting before the cells then
This was asked
that
in
were
lecture
infected
too,
go
though.
on to infect other cells.
This was
If you
actually
just had
asked
the
in
G
lecture too, though.
glycoprotein, then it would infect everything. You wouldn't know what
Like where it's coming
where
from,
it's
right?
coming
Like the
from.
source.
Exactly. Yeah, exactly.
Yeah. I agree, but
So actually, that was the same question was asked in lecture, and I think it was the student who was sitting right beside you asked that question in lecture, and it was the same question, like why didn't you just have that at the beginning? And I think I explained it in the same way. That in order for you to see like how this is spreading, like if you didn't know that it got into this cell first, how do you know that it didn't get into the vagus nerve first and is just going backwards? Like you wouldn't. You need to know that this cell is then transmitting to the next cell, which is the vagus, and that forms the rest of the circuit. If you just start to see it in both cells at the same time, then you wouldn't be able to tell, like, is it going retrogradely or is it like an anterograde? So you're just really being careful in confirming that these things are connected together. And again, this particular virus, the way that they've particularly engineered this virus, it does infect the neuroepithelial cells, but you want to make sure that it isn't infecting other cells. So you could add that layer as well. Okay. All right, as long as everyone's clear, and as long as you know that this RABG will form this missing G-glycoprotein, and that's why it's a delta G, it's missing this G-glycoprotein. These cells normally don't have that, so if we introduce it genetically into like another mouse or rat, a separate rat that contains this, then we'll be able to actually trace out the entire pathway, because now this virus becomes transsynaptic. It's not trapped in one cell. Okay, just to complete things off, this connection between this neuroendocrine cell or enteroendocrine cell or neuropod cell, like however you want to define it, actually does cause the release of glutamate. Okay, so the last slide that I'll get to is, if I can get to it. Oh, no, wait. I swear it did that all last lecture too.
With a couple of slides, yeah.
Okay, so phew. So do you need to know this slide? Yes, no, maybe?
Yes.
So I'm gonna ask you to kind of know the points that are up here at the top. You don't really need to worry about the ratios of the different enterotypes. This was just to show that there's a difference between individuals, age-matched controls, versus individuals who are depressed, that there actually is a significant shift in the enterotype. You can actually see, like on average, we are seeing differences in the bacterioids, and we're also seeing that there's a shift in some of the other types, like the ruminococcus as well, and these are the three that we saw earlier. These make and define what an enterotype is. And then if we do a closer examination of the individual types of bacteria, we see that we get different types of bacteria that actually, if you are overexpressing them, you actually feel better. These are associated with a higher quality of life. And the one thing that's missing in depression is Croprococcus. So Croprococcus, which is normally high in individuals that are feeling pretty good and have a good quality of life, that's completely gone or eliminated. or eliminated in individuals with depression. So those are the things that I'd like you to make sure that you know. Oh, please don't do that. How does that look online, by the way? Like, what does it do when it does this?
So right now, because Jeff has just set it up to directly stream your screen,
Right.
they can see the little,
Like this whole thing?
yeah,
So they see this whole thing.
yeah, because I think Jeff mentioned there was a little bit of an issue with the HDMI recognizing. I think your poor computer is just overheating.
My computer's overheating.
You can hear the fans going.
Okay, so while we're waiting for so it's like I have to literally for these courses, I literally have to buy like a new computer every year, which I hate doing because that happened the year before. So we bought this one last year, and that means that I'll probably have to buy another one for 2020. That's terrible. Yeah, but then I have to carry more stuff. Sorry to complain online, folks. So. Okay, all right. So the slide, one question, I'm kind of including it as sort of a giveaway question, but you do need to know stuff on this slide just to let you know, so that kind of hopefully gives you a little bit of time to start thinking about that. I decided for the October 9th term test, this will be like one of the questions that you're getting in advance. And this actually, if you're thinking about it, what slide do you think this matches up with?
The slide that you were talking about when Coprococcus?
Could be, or it could be, yeah, it could be, or it could be like related to this. Because a lot of this is related to like imaging data and brain scans. So again, think a little bit about that, and think a little bit about what you might expect to see. And then I give you sort of what we are increasingly trying to get you to think about, because it's actually a better type of question to get in advance if you're really studying, which is this. So again, I don't want there to be any surprises for you, but here we're looking at different changes in bacteria. And here's how I would want you to think about it. So Dialister is one that I've shown you related to depression, and so is Cropococcus. And we've also talked about things like Prevotella, and these two might be due to changes in mood states, so A and D. And Prevotella might be related to just like enterotype, but it might also be related to a change in diet. And then this one is kind of like completely different. Right? If you're thinking about it, you might be wondering, did we even talk about Staphylococcus? And in fact, if you have been studying, there is like one slide where Staphylococcus actually is very important early on, right? So you might want to start thinking about like, okay, how might I approach this? But these are the answer choices, and now you need to sort of figure out like, how confident am I in knowing what might happen in these different situations? Okay, now I'll just kind of open it up to questions. So yeah, did you have a question?
So these answers are different than for the question that you just showed us, correct?
These answer choices. So what I'm showing you is two different questions. So this is question number, let's say, 5, and this would be like question number 6. They're not connected in any way.
I see.
So I think if you are planning your study strategy, like you'll be at least familiar with, and these shouldn't be surprises for you at all. And if you go through some of the quizzes and everything else, you'll probably have like a good jump start on about 5 to 6 multiple choice test questions. How many of you are studying in groups? It's a group of 1. Yay, yay, yay. So I think if you can, like my hope is that you're going to spend the weekend kind of reviewing what you need to know. Lecture 1 is one that if you've been studying up until now, don't. Like go back over 2, 3, 4. Those are the more important ones. You kind of have an idea of like how to study for this type of question here. So reverse engineer that question. This one is a fairly easy one if you understand like what it's asking you. So it's telling you about MRI, brain imaging scans, and you probably need to go back and refer to this. And that's the way that I ask questions, so it kind of gives you an idea of like what to do. Plus, I happen to like Grey's Anatomy. So if you are studying, please do lecture 2, 3, and 4. Spend less time on lecture number 1. 3 is very dense. Lecture 3 is very dense. Lecture 4, there's a lot of memorization, so you need to be comfortable like in your studying, like which ones cause you more issues. So I think I'm going to spend like a couple of minutes if you have any questions, like just general questions. Sorry, I haven't heard from your colleague. Yep, you had your hand up.
Oh, yes. For the one with the, like, RAB-G, that slide,
Yeah.
that thing has, like, a graph of, like, CCK and, like, vesicular glutamate expression. Yeah, that guy, V.
Yep.
What's going on there?
So one of the things that they look at is if everything kind of matches up, these types of synapses are ones that are glutamatergic, and they're just confirming that these, in fact, do contain like some of the vesicular glutamate transport systems. But I didn't cover it, so you don't need to know, but just to follow up on you. Any other questions? So Yiwen, you had a question?
For the slide where you were talking about phosphoserine 129 alpha-synuclein antibody,
Yep.
you were talking about triton insoluble and triton soluble, and I was just a bit confused about your explanation. Could you please re-explain it?
What part of the?
It's on the slide that says gut microbiota required
Yep.
for alpha.
No, I know the slide, but what can I further explain for you?
So you would see phosphoserine 129 alpha-synuclein more staining for triton insoluble.
That's correct. So if we were to take the biochemical fraction, and then we were to see where we would have more phosphoserine 129, it would be in the fraction that has insoluble proteins.
Yeah. Because we're looking at Lewy bodies when we're using phosphoserine 129, and Lewy bodies are
Right.
insoluble.
Right. Correct.
Okay.
Right.
One thing you said in lecture that, like, I had to write down was that 90% of the alpha-synuclein in Lewy bodies is phosphorylated. So when you're finding alpha-synuclein that's phosphorylated, that probably means it's in a Lewy body. And so at that point, you find the phospho in the insoluble because it's a body.
Lauren,
Could you repeat that?
Yeah, 90% of alpha-synuclein
I don't even remember
in
saying
Lewy
that.
bodies is phosphorylated.
Huh. Okay. Should you write it down? I don't know. Sure, why not?
Thank you. So
Yeah, Lauren?
the test starts at 3:10?
4:10. Yep. It's like the regular class time. It just, I think I posted the wrong time. Why? Is it still on there at 3:10? Like we're
Like, I saw the email and I was like, oh, okay, gotta get
Yeah,
there.
No,
it's
but-
just
but-
like when you got the notification, because they
But-
can't go back and change the.
But I think on the announcements page it has like 4:10. Yeah, I forget that it gets emailed to you guys. No, is that still okay for you, Lauren? 4:10?
That's better because we were at 3:10 and I'm not sure if I can make it.
Right. No, it's the regular class time. So 4:10. Any other questions? Are there any questions online? Any other discussion questions that I've missed?
No, there were only 3.
There are only 3. Oh no. Wait, how does this work when you study at home? Do you guys study separately? Do you study together? I'm quite curious.
Separately usually, and then come together for like questions.
Ah, interesting.
Because you study out loud, so it's pretty annoying.
Oh.
Hear
Okay.
each other.
And Michael, are there any questions online? None.
They're satisfied.
They are satisfied. How do you know they are satisfied?
I we have two likes on the video.
Wow, wow, wow. So wow, so I'm,
You're on fire.
so those are my suggestions for you to study, like how I tackle 2, 3, and 4. If you're comfortable with everything now, the only other thing that I would do is, as you study over the weekend, if there are any small details that you need clarification on, or you just want to come in and chat about them, for Tuesday, it's probably a good idea to make an appointment. For Wednesday, it's just like kind of open. You can drop in, you can ask a question if that's all you need to do, and then you can just leave, or you can decide to study there. We want to keep it to a fairly quiet space, so it's actually on the opposite side of the stairs. You actually have to go down a small hallway, and then it used to be like the principal's residence in New College, but it's pretty quiet, and we want to keep it as a quiet space. So if we have questions, we'll kind of just step out, answer them, and come back. And we can also use the screen that we have there. I'm expecting you guys to do well. That's the expectation, so I'm hoping that everyone does do well. I'm not sure if we're going to have bonus marks this year. We did last year.
That would be wonderful.
Like for what? An extra question or?
Yeah, it's like bonus questions are always optional. Like you don't lose marks if you try them. So don't know, still debating. I'll leave it up to Farbad. Farbad, will you have it? Will you mark them all for me too then?
Yes.
Okay.
Does that mean he gets to decide what the bonus question
No, no, no. Last year, I have to tell you, the bonus question was one that was on the slide that I actually covered, but most students were like, did we cover that? And it was, what is the ganglia that is associated with this particular pathway? And
Node
it,
cell.
and it turns out to be the nodose ganglia.
Those, yeah,
And students were like,
Not the bonus question.
no, no. Last year we actually had 2 bonus questions.
Oh, that one.
Right, that one. No, but last year I actually covered it, and I mentioned like, oh, this is sort of the group of cell bodies. Remember the ganglia that you learned about in PSL 300? And a lot of students were like, are you sure we learned that? And I said, yes, you did. And I think that bonus question, like 10% of the class got it. But it was a bonus question, so there's no harm in doing that. So for Bod, you say yes, and you're going to mark all of the...
I will mark all of them, yes.
Okay.
Excluding mine, just.
Okay. Well, that's fair.
Why would you exclude your own?
That's fair. That's fair. Okay. So how many bonus questions should we have then for Bod, since you're going to mark them all?
How many students are there in the class?
I don't know how many are actually going to write on Wednesday. I'm hoping most of the class does. It's a very fair term test, and we have 310, so we're over enrolled, but I'm thinking that probably 270 will probably write on Wednesday. I'm hoping. I don't know. Like we're leaving it open for students, and some students are they're just feeling swamped right now. So how many? 2? 2. Yeah, that's how many we had last year. Yeah, 2. 600. Yes. Okay. For Bod, you also have to remember someone has to input that into like the mark, and Quirkus actually doesn't allow you to give you more than, like if it's out of 30 and you set the mark out of 30, you can't actually get like 31 out of 30. You have to reset the whole thing. So whatever you get on your term test, it's like out of 30. I think, right? So it's 10. Yeah, it's like 20 multiple choice, 5, and then 5. So it's out of 30. And then your bonus marks would be like half a mark each. So it'd be like you could get up to 31 if you got both bonus questions.
Where do you put that extra mark?
I'm not sure. Last year we just kept like a separate spreadsheet, and then we added the mark back. I'm not sure how that works. It doesn't work well on Quirkus unless they fix it. Last year we couldn't, like when we set the mark, you know how you set an assignment and it's worth like 20? You can't actually give more than 20. At least not last year. We did complain about that, and they were like, "How could you get more than the mark?" So we kind of tried to explain.
I think it's still similar, because I had other courses where the bonus mark, they would just make a separate, like, grade
Yeah,
section. and it
so-
was like a
So it's a weird thing about Quirkus that we don't get. So Quirkus, you can't actually post on discussions like anonymously, which is very troubling for some students. So it's not a perfect system. So 2 bonus questions. I'll have to write that announcement.
We didn't get any bonus questions for CJH.
No,
No, we didn't.
no.
Well, thank you.
All right. Well, you get them now in 300.
But now I get to one.
Yeah, right, exactly. Okay, all right. Okay, 2 bonus questions. All right, awesome.
One each.
Okay, so we're done. Have a good weekend, everyone. Study. Take it easy, though. Like, more important than like studying is just making sure that you're taking care of each other. Your mental health is really important.